So I'm
Yes.
very excited to introduce Leandros Boukas, who is now a pediatric resident at Children's National Hospital in Washington, D.C. He's actually going to be starting his medical fellowship at Harvard Medical School later this year. So Leandros has been performing both his PhD as well as postdoc researches in Johns Hopkins School of Medicine. Its research has mainly focused on the mutation rate as well as methylation of CPG islands, along with other epigenetic markers in human disorders. And he has won numerous awards as well as recognition for excellence in terms of presentations, such as Reviewer's Choice Awards, as well as Fellowship for Academia Excellence in the US. Now, I'm very delighted to welcome him today as he shares his insights on leveraging Mendelian disorders of the epigenetic machinery to characterize functional epigenetic changes across cell types.
Okay, thank you so much for this kind introduction. I really am delighted to be here. This has been one of my favorite seminar series for several years. And so I'm gonna share with you some of the things we have learned together with Hans Bjornsson and Kasper Hansen by studying this new class of Mendelian monogenic disorders, which are called the Mendelian disorders of the epigenetic machinery, and as their name suggests, are caused by loss of function mutations in genes encoding epigenetic regulators, and as such can provide a really unique window into the causal role of epigenetic changes in disease outside the context of cancer. And so my talk is going to be centered around three questions. The first one is, given that those are genetic disorders with epigenetic consequences at the root of their pathogenesis, we want to know what the nature of those epigenetic consequences is, meaning how many locations along the genome have disrupted accessibility, what is the magnitude of this disruption at the individual locations, and in what direction does accessibility change? Does chromatin become more open or more compact as the result of the genetic mutations? The second question is whether it's the same locations in the genome that preferentially are disrupted between the different, when we compare different disorders to one another. And this is especially interesting in light of the fact that even though each of these Mendelian disorders is caused by a loss of function mutations in a different gene, there are several shared phenotypic manifestations. And so we are wondering whether this convergence at the phenotypic level can be explained by convergence at the epigenomic level downstream of the distinct genetic defects. And then finally, we want to know whether different cell types have a similar chromatin disruption or whether the chromatin disruption is different between different cell types. And just to give a little bit more context here, we had previously shown when we did a comprehensive analysis of all human epigenetic machinery genes that these genes are expressed essentially in all cell types in the human body. And so this begs the question, if two cell types both express the same mutant gene, then is the ensuing chromatin disruption the same between those cell types, or is it different? In order to answer those questions, we use mouse models of three disorders of the epigenetic machinery: Kabuki syndrome type 1, which is caused by haploinsufficiency of KMT2D, Kabuki syndrome type 2, which is caused by haploinsufficiency of KDM6A, and Rubinstein-Tabi syndrome type 1, which is caused by haploinsufficiency of CREB binding protein. I will mostly focus on the two types of Kabuki, but I will occasionally mention some comparisons with Rubinstein-Tabi as well. And I will also mention here that all three of these genes encode transcriptional co-activators, histone methyltransferase, a histone demethylase, and a histone acetyltransferase, respectively. And then we looked at three cell types which we knew were involved in the disease process: B cells, T cells, both of those are immune cell types, of course, and then also neurons from the dentate gyrus of the hippocampus. And in B and T cells, we did a tag sequencing to profile chromatin accessibility, as well as RNA-seq to profile expression, whereas in neurons, for technical reasons, we're only able to do a tag-seq. It's important to mention here that chromatin and RNA were always isolated from the same mouse and at the same time point to ensure a robust comparison. And then also, in order to ensure that we had a well-powered experiment, we always had at least 5 mice per genotype. So the first question, as I mentioned, is what is the nature of the epigenetic changes in these disorders? So here, the x-axis is grouping the results by cell type. And then within each cell type, we're looking at Kabuki syndrome type 1 or KS1 and Kabuki type 2. And the y-axis is showing the fraction of attack peaks or regulatory elements at promoters that have disrupted accessibility in the mutants compared to the wild type. And the take-home message from this plot is that there is a widespread disruption of chromatin accessibility along the genome. In neurons, it's about 70% of promoter regulatory elements that are disrupted. In B cells, it's about 20%, and in T cells, it's about 10%. And even that 10% corresponds to more than 1,000 promoter regulatory elements. And then when we look at the magnitude and the direction of this accessibility disruption, here the x-axis is showing the log-fold change in chromatin accessibility with a positive value indicating chromatin shifted towards a more open state in the mutants compared to the wild type. And then the y-axis is again grouping the results by cell type, and within each cell type, we're looking at Kabuki type 1 and Kabuki type 2. And we can see that in the overwhelming majority of cases, chromatin becomes more open in the mutants. And also, the vast majority of the chromatin accessibility changes are of small magnitude, almost always less than twofold. So then the next question is whether it's the same locations preferentially disrupted in the different disorders. So to tackle that question, we actually had to develop a new computational method. The reason being that the traditional approach, which would simply consist of taking a list of regulatory elements disrupted in one condition, say Kabuki type 1, taking a list of regulatory elements disrupted in another condition, and then looking at the intersection of those lists, this approach generally has very low statistical power, and it is especially ill-suited for the problem that we are interested in here because we have multiple changes which are individually of small magnitude. Instead, the approach that we developed works as follows. We have two comparisons, say Kabuki type 1 versus wild type and Kabuki type 2 versus wild type. And from each of these comparisons, we are testing the same set of attack peaks or regulatory elements for disrupted accessibility in the mutants compared to the wild type. And what we can do is we can take those regulatory elements disrupted in Kabuki type 1, and then we can look at their p-values in Kabuki type 2. And then we can compare these p-values to the p-values of those regulatory elements that are not disrupted in Kabuki type 1. And then if it's the same locations that are preferentially disrupted more than expected by chance, then the green distribution here, which corresponds to those regulatory elements disrupted in Kabuki type 1, it's going to have this spike at 0 in Kabuki type 2. Whereas the black distribution corresponding to the null elements is not going to show this spike, or if the spike is present, is going to be much less pronounced. So we show that this method actually has much greater power and also lends itself to a easy visual interpretation, as I'm showing here. In contrast, if the two conditions do not have the same locations disrupted more than we would expect by chance alone, then the two distributions will be identical to one another, essentially. So we applied our method to the actual data. Here I'm showing all the cell types, and then the red distributions are the p-values in Kabuki type 2 of those regulatory elements that are disrupted in Kabuki type 1. And what we can immediately appreciate is that those red distributions have a much more pronounced spike at 0 compared to the blue distributions, which correspond to the elements that are not disrupted in Kabuki type 1, which indicates unambiguously that it's the same regulatory elements that are preferentially disrupted in Kabuki type 1 and Kabuki type 2 across the cell types. And of course, we want to know, as I mentioned, whether this chromatin disruption is related to the phenotypes, with an obvious first step being to look at gene expression. And in fact, we show that most of these changes are also associated with gene expression changes. We quantify this genome-wide in the papers, but here I'm just showing an individual example where we're looking at this gene part 3B, which is a regulator of cell polarity. And then we are looking at its promoter at the chromatin accessibility of its promoter, with orange lines depicting the accessibility signal of Kabuki type 1 mutants, the green and the pink lines depicting the accessibility signal of the Kabuki type 2 and Rubinstein-Tabby mutants respectively, and the black lines depicting the accessibility signal of the wild type mice. And then we are also looking at the corresponding change in gene expression with the same color coding. And we can appreciate that chromatin is shifted towards a more open state in the mutants, and expression is upregulated in the mutants. And also, if we focus at Kabuki type 1 and Kabuki type 2, we can see that there is a quantitative correspondence in the sense that the fact that Kabuki type 1 mice have a greater shift towards open chromatin compared to the Kabuki type 2 mice translates into greater expression upregulation. And we show that this correspondence is in fact the norm genome-wide when we look at gene and promoter pairs that are both disrupted. And there's ongoing work in the lab led by Leon Di Stefano, where we are actually further developing this method so that it can robustly tackle comparisons of multiple disorders at once. Here, I don't have time to go into the details, but I will just mention that this spike at 0 indicates that it's the same genes that are dysregulated between the different disorders in terms of their expression. And in fact, a gene set that contributes to this spike here is transcription factor genes, which may explain why individuals with these Mendelian disorders have been shown to have abnormalities in B cell maturation and development, which then lead to infection susceptibilities. And then finally, the last question is whether the different cell types exhibit preferential chromatin accessibility changes at the same locations. And so again, we applied our method in order to compare B cells to T cells and B cells to neurons. So here the red distributions correspond to those locations that are or those regulatory elements that are disrupted in B cells. And we are looking at their p-values either in neurons or T cells, in Kabuki type 1 on the left and Kabuki type 2 on the right. And what we can appreciate is that the same locations are preferentially disrupted in B and T cells, but this is not true when we compare B cells to neurons. As we can tell by the fact that the p-value distributions are essentially identical. And what is, I think, really remarkable about this is that this is not explained by the fact that the chromatin accessibility disruption in neurons is localized at locations that are only active in neurons and not in the other cell types. So here the pink bars depict regulatory elements that are active in both neurons and B or T cells, whereas the dark gray bars depict regulatory elements only active in neurons. And the y-axis is showing the fraction of peaks that have disrupted accessibility. And so we can see that this fraction is always greater when we look at peaks that are actually active across cell types, with the exception of KS2 promoters. And so then we wanted to ask what could be some of those locations that are constitutively active and are only disrupted in neurons. And we looked at CPG islands, which were a good candidate fitting this criterion. And so here on the x-axis, I am grouping regulatory elements into deciles, with the first decile containing those regulatory elements for which we have the strongest evidence of disruption, and the 10th decile containing regulatory elements for which we have essentially no evidence about disruption in the mutants. And the y-axis is showing the fraction of regulatory elements that overlap promoter CPG islands. And what we can see is that in neurons, but not in B or T cells, the fraction of regulatory elements that overlap CPG islands is much, much greater when we look at those that are disrupted. And it's only a few of those regulatory elements that fall within CPG islands when we focus on those that are not disrupted. And then this is showing that the same preferential localization of the accessibility disruption at CPG islands is present in Kabuki type 2 as well, in neurons only and not in B or T cells. And then finally, an interesting observation that was a follow-up from this result is that some of those CPG islands in fact correspond to other epigenetic machinery genes. For instance, here I'm showing that the CPG island at the promoter of the gene that's causing KS2 is disrupted in KS1, and the CPG island of at the promoter of the gene that's causing Kabuki type 1 is disrupted in KS2. And we expanded this observation in the paper to show that the epigenetic machinery gene promoters are preferentially disrupted in neurons, but not in B or T cells. And so this is a summary of the results along with some open questions. Haploinsufficiency of those epigenetic machinery genes causes widespread chromatin accessibility changes that are individually of small magnitude. The same locations are preferentially disrupted in the different disorders, and the same genes are preferentially dysregulated, which might explain some of the shared phenotypic manifestations. And there are neuron-specific accessibility changes at locations that are otherwise that are active ubiquitously across cell types, such as CPG islands. And of course, some open questions have to do with how many of those small changes actually have a causal contribution to the disease phenotypes. How small can a change be and still have a discernible phenotypic effect? What explains the bias towards increased open chromatin at promoters, even though the loss of function mutations disrupt transcriptional co-activators? And then what is the mechanistic basis of the neuron-specific changes at locations that are constitutively active? So I want to thank Hans, Casper, and Teresa, with whom we did this work. Here are some of the papers. And here's a little database where we have organized all human epigenetic machinery genes. And I'll be happy to take questions. Thank you for your attention.
Thank you. If you have any questions, please feel free to type it in the Q&A box or raise your hand. And I guess the KMT2D or the KDM6A are in such a large complex. Why just these two proteins? Have you found other drivers of the syndrome? Like why these two?
You mean if we have expanded the comparisons beyond just Kabuki type 1 and Kabuki type 2?
Yeah.
Yes, so we have looked at Rubinstein-Taybi syndrome, which is caused by haploinsufficiency of CREB binding protein, which is a histone acetyltransferase. So this is not, as far as I'm aware, has not been shown to be part of the same complex. And yet we see that even when we compare Rubinstein-Taybi to the two types of Kabuki, the same locations are preferentially disrupted.
Mm-hmm. So someone asked in the chat box, the nanopore and the targeted MLL. No, this is different. Sorry, this is a different question.
No worries.
I think it's also 1:05, so we are 5 minutes about over time. So maybe we can just stop the seminar here. If you have any questions, I think you can email the speakers. I'm sure they will be very happy
Can I ask
to answer
a quick question?
Yeah.
Can I ask
Thank
a quick
you
question?
all for the wonderful presentation. Yeah. Go ahead,
if people want to leave, if people want to leave, they can go ahead and drop out. But to follow up on your question from your conclusion slide, do you have any speculation as to like the mechanism through which, getting rid of or a haploinsufficiency in a K4 trimethylase or in what's demethylating K27 is causing increase, or losing those activities is causing an increase in accessibility?
Yeah, so this is a question that we've been thinking about a lot, and we have some hypotheses at this point. So, I mean, one hypothesis is that some of those changes are actually indirect consequences, right? So not all of these changes need to be direct consequences of the haploinsufficiency, because you could have some direct targets and some of those might actually be repressors. So then losing the repressors might lead to downstream shift towards open chromatin. Another hypothesis is that there's other proteins which somehow overcompensate when you lose those, and this causes a compensatory shift towards more open chromatin that exceeds what the normal balance that you would normally have. And then I actually just read the paper that you published, so I'm actually very interested in thinking more about whether this connection with lack of transcription and histone deacetylase, deacetylases, which ends up shifting the balance towards more open chromatin.
Cool. Have you been able to do any like acute experiments to take wild type cells and like briefly reduce or knock down these proteins?
Not yet, but I think this using something like an auxin-inducible Degron technology or something like that would be a great way to try to sort of have a more time-resolved picture of how the different defects arise.
Awesome. Well, thanks so much. We like Sky said, we don't want to go way too far over time in case people are trying to get to another meeting. So Sky, Ultus, thanks for hosting. Leandros, Roberta, and Emmanuel, thanks for 3 great talks. And anyone still in the room, if you or any of your trainees or any of your friends are interested in giving a talk, our application form is still open, and we will be looking for additional volunteers to give talks in the fall. So please feel free to pass that link along. You can find it in our emails or on Blue Sky. And I hope everyone has a wonderful rest of their day.
Thank you so much.
Thank you all for presenting.